Hello dear viewers, welcome to Mr.Zoologist YouTube channel. Myself Nasir Khan, and today we are going to discuss about cell cycle clock. The cell cycle clock is a network of interacting proteins, a signal processing circuit that receives the signals from various sources both outside and inside the cell, integrates them and decides these cells' fate. So this protein network decides the fate of the individual cells throughout the body. Thus, almost all type of normal cells will not proliferate unless prompted, stimulated to do so by mitogenic growth factors. Some other signaling proteins like transforming growth factor TGF beta may overall cancel the messages conveyed by the mitogenic growth factors and force a halt to proliferation. The cell will stop the progress from the different stages of cell cycle and will enter into a resting phase or GO. So extracellular signals may persuade or convince a cell to enter into a post-mitotic differentiated state from which it will never re-emerge to resume proliferation. So in some way, this mixture of signals must be processed, integrated, and ultimately a clear decision is made by the cell as to whether it should proliferate or become quiescent, and whether as a quiescent cell it will or will not differentiate. These behaviors suggest the existence of some central acting governor that operates inside the cell that receives a wide variety of incoming signals and makes major decisions concerning the fate of the cell. So this master governor has been identified. It is a cell cycle clock which operates in the cell nucleus. Should the cell cycle clock decides in favor of proliferation or continuing towards the cell cycle, it proceeds to orchestrate or arrange the complex transition that together constitute the cell cycle of growth and division. Should it decide in favor of quiescence, it will use its agents to impose this non-proliferative state on the cell cycle, will keep the cell in the resting phase. So these are the different factors controlling the cell cycle clock, such as growth factor receptors, mitogenic monitors of genome integrity, TGF beta transforming growth factor beta receptors, integrins, and monitors of cell cycle metabolism. The cell cycle clock will either decide programming of cell cycle phases, such as to allow the cell to proceed through the G1 to S, G2 to M transitions, or will lead the cell to enter into the GO or quiescent state. So the term cell cycle clock denotes a molecular circuitry operating in the cell nucleus. It proceeds and integrates a variety of incoming signals originating from outside inside the cell and decides whether or not the cell should enter into the active cell cycle or rest into a non-proliferative state. So in this event that active proliferation is decided upon, the cell cycle clock proceeds to program the sequence of biochemical changes in a cell that enable it to double its contents and to divide into two daughter cells. The proliferative behavior of cancer cells indicates that the master governor of the cell's fate is influenced not only by normal proteins but also by oncogene proteins. These proteins insert themselves into various signaling pathways and disrupt normal control mechanisms. Similarly, the decision of key tumor suppressor proteins evokes equally profound changes in the control circuitry. This deletion is very effective in perturbing decision making by the cell cycle clock, and hence it may decide in favor of uncontrolled proliferation or quiescence. So that was the video for today. We will meet in the next video. Till then, goodbye.